In our previous sessions, we've understood the structure of the DNA, the different experiments that led to this discovery of the structure of the DNA. Also, we've learned about the process of replication of the genetic material, which is the DNA. In today's session, we will learn about the next important aspect of genetics or molecular biology, which is transcription. And we will understand how it basically happens in prokaryotes and eukaryotes. Now, here in this particular diagram, we have something called as this is basically the representation of the central dogma of life. And what does the central dogma of life tell us? It tells us that DNA is basically the genetic material of all living organisms, most of the living organisms, barring a couple of viruses. And this DNA is then transcribed. Transcription basically means to convert the script. So we've got a script which is converted into another script. So let us say you cannot understand you don't understand German, and so you bring in a translator and you ask him to convert the German script into English, and this process of translation or basically conversion of a script of German into English is called transcription. So basically making something which is unreadable into a readable content is called transcription. So the cellular machinery cannot read DNA directly. DNA cannot be read directly. So DNA is converted into a particular molecule, which can then be read by the various molecules or machines of the cell in order to generate proteins. This process here is called transcription, to change the script. So central dogma says the DNA is converted into RNA, and then the RNA is read by the different molecules or components present within the cell in such a way that a protein, the information about the protein is obtained and a protein is synthesized. This is the central dogma of life. Transcription leads to translation. Now, the interesting point here is, why has life evolved such a complicated mechanism? Why DNA directly cannot be used to synthesize proteins? Why DNA can directly not be used to synthesize protein? Now, the first thing that we must bear in mind is that the primitive, the older genetic material is the RNA, and it has been established by a couple of experiments and a couple of theories that we have at our hands. So RNA was the first genetic material which eventually evolved to give rise to DNA. Right, now that is the first point that we must bear in mind. But why DNA directly cannot be used to make proteins? Why do we have to undergo these particular processes, spend a lot of energy, generate a lot of different types of molecules? Let us just investigate a little bit of our time into it. Let us invest our time into it and investigate this particular issue. Now, first of all, think about this. Even the smallest of the DNA is extremely longer and very large compared to a segment of RNA, a small RNA. Let us say you compare an mRNA molecule and compare it to any particular DNA molecule. Most of the molecules of DNA will be extremely long and intricate. In prokaryotes, the DNA is circular. In eukaryotes, it is extremely long, highly coiled, packed in the form of chromosomes, but it is not circular. That is one thing you must remember, but it is extremely highly coiled, tightly packed, very long. So if DNA was to be used as a source of a protein, the opening up of DNA would have become extremely frequent. And opening up the DNA will take a lot of energy. You have to, for example, this particular sequence of the DNA makes up, which is an extremely long segment here, but let us say this has to be used to make up a certain protein. So this DNA would be opened up like this for the synthesis of the protein. Let us just assume for a moment that the cell can read these particular sequences, and for making a protein, the machinery will first of all run through it to make a protein once, and then we'll run through it again twice, and third time, fourth time. So the strand will have to be kept open, and a large section and wide variety of this protein, of this DNA segment will have to be kept open, and this would require a lot of energy. So energetically, this is not a very favorable proposition, not a very favorable idea, and that's why probably life has not investigated or investigated this route of synthesis of protein. Another possible reason is that let us say DNA intends to replicate. Let us just take a prokaryotic DNA, circular DNA. Let us say it intends to now replicate. So we will have a replication bubble, and then the two strands will separate out. But you must bear in mind that for all of these replication processes, we also require a wide variety of proteins. Now, you've only read a handful of enzymes that are involved in the replication of DNA, or you have read only a couple of proteins, but there are a large variety of proteins required. Now, while the replication of DNA is going on, if the DNA itself starts to make proteins, let us say it is making proteins while it is also getting replicated, the DNA polymerase enzyme and the other components will encounter these sites from which are now being used to synthesize protein. So there will be this interruption, there will be basically a collision of this DNA polymerase enzyme with the site which is producing the protein. So again, that is not a very good idea to have to make proteins while they are replicating. So how has the cell overcome this? So what the cell does is, out of its genome, it already makes large number of RNAs, which are then kept probably in the Golgi body or in some other structures temporarily. And when the DNA is replicating, these stored mRNAs or let us say dormant mRNA strands are then eventually become active. So while the process of replication goes uninterrupted, these mRNAs are also synthesizing protein molecules which are required for the process of replication. Now, another reason could be that protein making systems, we call them the ribosomes. Now, ribosomes have evolved in such a way that they are great at reading mRNA strands. Even there are other type of RNA molecules which we will study soon. For example, the tRNA molecule. All of these are protein making components, the tRNA, the ribosomes, etc. All of these molecules are designed or have evolved in such a way that they are great at reading the information present in mRNA, but they have not evolved fairly well to understand what is encoded in the DNA. So this is another reason why DNA cannot be used directly for the synthesis of protein. Another problem can be, let us say now talk about the eukaryotic cell. So we have got the nucleus, we've got the cell here. So let us say the cell intends to make its own make some certain proteins, and this is the DNA. So think about this: if this eukaryotic cell has to make certain proteins, where are the proteins made? They are made in the endoplasmic reticulum, the rough endoplasmic reticulum, where we have the ribosomes. Now, every time a protein has to be synthesized, the entire DNA molecule will have to be transported out to the nuclear. Synthesis of the protein have to be done, and then this has to be brought back. And so this is actually an impractical idea. And for these particular reasons, the DNA never evolved. Because of so many problems with DNA never evolved as a molecule that could directly be used for the process of transcription. RNA, on the other hand, provides a lot of comfort, at least with respect to these points, and there are many other points. So let us proceed further and try to learn a little bit more about transcription. So what is transcription? Let us come back, as we just saw it earlier. So transcription is the synthesis of RNA from DNA template. So we've got this DNA double helix, and it has got its own base pairs, variety of base pairs. Now there is an information about the synthesis of a particular protein or about certain other molecules we call RNAs hidden inside them, and in order to get that information out, this DNA strand is split open and it is read by certain molecules, and an mRNA, not mRNA, let us not specify first, let us just put a general word, and RNA molecules are synthesized. So this process of converting the information present in DNA into a more readable RNA molecule, this process is called as transcription. Now, why did I not use the term mRNA for transcription? Because mRNA is just one of the RNA molecules that basically is generated out of these DNAs. So the DNA molecule basically generates three different types of RNA molecules. The first type of RNA molecule is known as the messenger RNA. The other type of RNA molecule is known as the ribosomal RNA, and the third type of the RNA molecule is called the tRNA. Now there are many other types of RNA molecules, but these are the three major classes, three major types of RNA molecules. Now the messenger RNA, mRNA, as you can see in this image here, is a linear molecule. It is a single-stranded molecule with a lot of base pairs on it. Now you must remember, as these are RNA molecules, they will have a uracil in place of thiamine. Right. So what do we have here? The messenger RNA is a linear molecule, and its function basically is basically it contains the information regarding the sequence of amino acids that will make up a particular protein. So basically these three bases here contain the information regarding a particular amino acid. Let us say they encode for an amino acid called arginine, and the next three they code for something called as cysteine, and because they are adjacent together, they also contain the information that these two amino acids must be bound together, and similarly many other amino acids must be bound together. So this is the information that an mRNA carries. mRNA molecule, messenger RNA, basically carries the information about the sequence of amino acids present in a given protein. The second type of the RNA is known as the ribosomal RNA. Now, you have in your junior classes, in your younger classes, have studied about this excuse me, molecule called the ribosome. Now, ribosome is primarily present on the surface of the endoplasmic reticulum, where we call it the rough endoplasmic reticulum in eukaryotes. In prokaryotes, ribosomes are found floating in the cytoplasm, freely floating. Now, ribosome, the entire this whole structure is the ribosome. Ribosome is made up of two components: the protein component, the protein part, and the RNA part. So both of these macromolecules come together in a certain specified configuration and then give rise to a ribosome. Now, ribosomal RNAs are also synthesized from the DNA. DNA also contains information about the sequence of these RNAs, or the information about the segments called ribosomal RNAs. And these ribosomal RNAs are extremely important in making ribosomes. So ribosomal RNAs are also synthesized, or also transcribed from the DNA. The third type of RNA that is transcribed is known as the tRNA, and let me just show you where the tRNA is. So this part here, this part is the tRNA. This is the tRNA. This bluer structure on the top is an amino acid. So what is tRNA? tRNA stands for transfer RNA, and the function of transfer RNA is to bring an amino acid next to the other. And we will study how this happens in our next session when we study the process of translation. But at the moment we are only going to bother ourselves with transcription. So this is basically the transfer RNA or tRNA. I hope you've understood the definition of transcription. And transcription, once again, let us just go through it, is the synthesis of RNA molecule from the DNA template. Now it can be any kind of the RNA. It can be the mRNA, the rRNA, or the tRNA. Before we proceed any further, let us look at a couple of terms and understand them pretty well. Now there are these three terms. The first is the gene, the second is what is a cistron, and then we'll understand these two terms as polycistronic and monocistronic mRNA, and we'll also understand the different types of genes. Now, what is a gene? A gene is a segment of DNA that contains information about proteins or the various types of RNA molecules. Now let's just get a little bit of space. So what I mean to say is, let us say this is our DNA, and I'm not going to draw a helix, and this is the ladder-like structure, and we've got the two anti-parallel complementary strands. So let's say this is the 5' 3' and 5' 3' anti-parallel strands. Now the entire DNA is extremely long, right? If you look at the chromosomes of the mammals, of the humans, or of any other particular cell, the DNA is extremely long. The entire DNA does not contain the information about the sequence of amino acid in a particular protein. The entire DNA does not contain information about the sequence of bases in the different types of RNA molecules. Most of the DNA is basically non-functional. It is non-functional with respect to the presence of information inside it. Most of the DNA does not contain information inside it. But the parts of the DNA, let's say this is the huge massive DNA, just fictitiously thinking. Now, let us say this particular region, this region here, and this region here, these regions encode for either proteins, or let's say they encode for proteins, or they encode for some kind of an RNA molecule, maybe an mRNA, rRNA, or tRNA. These segments that either code for RNA molecules or code for proteins are known as genes or a gene. So gene is a segment of DNA that contains information regarding a protein or regarding various molecule types of RNA molecules. Now, what is a cistron? A cistron primarily is a segment of DNA. Again, it's a segment of DNA. A cistron is also a segment of DNA. But now let us again draw the same kind of a molecule. So let us say we have the DNA here, and I'm going to use a green color to show. So these green areas are basically regions that will only make up the RNAs. So all of these green regions will only give rise to, so green region is only segments of DNA that will encode for some kind of an RNA, maybe a ribosomal RNA, maybe a tRNA, or just these two. But now let me just take a yellow marker here, and let us say this segment here and this segment here, these segments, they give rise to, the yellow segments give rise to mRNA. And the information that mRNA encodes for is basically the sequence of amino acids and hence proteins. So cistrons are the segments, the sequences on the DNA which carry information about the protein, which carry the sequence of amino acid about the amino acids that are present in the proteins. Simply put, the cistron is the segment of DNA which will give rise to mRNA. Right? So gene basically is a broader term. A gene includes cistrons also, which means mRNA coding sequences. Gene also includes the non-protein coding sequences of the DNA, which are basically ribosomal RNA and tRNA containing information. So I hope you've understood the difference between a gene and a cistron. Now, prokaryote, the mRNA molecule that is synthesized by prokaryotes is polycistronic. Let us just try to understand what do we mean by this particular term. So prokaryotes are polycistronic, whereas eukaryotes are monocistronic. Let's magnify and look into this image in order to understand it better. So here, what we have is, here I've got an eukaryotic mRNA. Now in this eukaryotic mRNA, there are a wide variety of, there are different sequences. We have got something called as a UTR, which you do not need to understand. Their translation starts right, you don't have to know this. But if you look at the center of this particular mRNA, you see that it is basically this one region is encoding for only one type of a protein. So what you must bear in mind is that the mRNAs that are produced in eukaryotic cells, one mRNA will encode for only one type of a protein, and such an mRNA molecule is called a monocistronic. So this is called a monocistronic mRNA. A polycistronic mRNA is a hallmark of prokaryotes. Remember, eukaryotes do not have a polycistronic RNA. They don't have it. Prokaryotes, on the other hand, only have polycistronic RNA. What do we mean by polycistronic mRNA? A polycistronic mRNA is an mRNA. Let's just drag the image slightly to the top. Here you can see it far more clearly. The prokaryotic mRNA, this is just one mRNA strand, and this particular strand contains information regarding the synthesis of more than one proteins. It's not just one protein, but it contains information. It basically has the code for 3 different proteins. Poly means many. So in a polycistronic mRNA, you will have at least the information about 2 proteins. There usually are more than 2 information regarding more than 2 proteins involved in a prokaryotic mRNA. Prokaryotic mRNAs are known as polycistronic mRNAs. Let's just demagnify it, bring the slide again back to the center where we can comfortably see it. Okay, now I hope you've understood the meaning of the term polycistronic and monocistronic. Monocistronic means only one cistron is present. One cistron means only one protein coding sequence, and polycistronic is many protein coding sequences. So we got 1, 2, and 3. Right. So I hope all of this is clear to you. Now we'll go to the next part, which is what are the different types of genes? There are 2 types of genes, broadly speaking. This is just one way of classifying genes. There are many other ways of classifying genes. According to this particular, according to one of the ways, genes can be classified into 2 types, which is basically the structural genes and the regulatory genes. Now, what are the basis of this classification is basically what kind of a protein is synthesized. Now a structural gene can synthesize three components. A structural gene can synthesize any of the RNA molecules. It can synthesize a gene is called structural when it synthesizes for a ribosomal RNA, or if it synthesizes a tRNA, or it synthesizes an mRNA, right, which has no role to play in controlling another gene. So what I mean to say is a structural gene, if it can either produce an rRNA molecule, it can produce a tRNA molecule, or it can produce mRNA molecules. What kind of an mRNA molecules? The proteins of which have no role in controlling other genes. We'll just have a look into it. Okay, so such kind of genes are called structural genes. And what is the other kind of a gene? The other type of a gene is called a regulatory gene, or are called regulatory genes. And what are regulatory genes? Regulatory genes make mRNA molecules, and the products or the proteins that these particular genes, regulatory genes, eventually make up, these proteins have one function only, which is to control other genes. Now let me take an example. Let me give you an example. Now there's this let's say there's this sequence of DNA. There's this DNA sequence, and let us say out here let's say this is the gene for the production of a hormone called insulin. Now insulin is required for glucose metabolism, for carbohydrate metabolism. Now this particular gene here is a structural gene because it produces insulin, and insulin controls the amount of sugar that is present inside our body, right? So it's a structural gene. However, let us say there is, now how much insulin should the body make? Should it make let us say 100 milligrams per 20 kilograms of the body mass, or should it make more, or should it make less, or does it always make the constant amount? So the interesting thing to know is that the amount of insulin produced by the body keeps varying. Sometimes it is high, sometimes it's low, sometimes it is kept at a moderate level. So it keeps changing. Now what is controlling the activity of this gene? So this gene will become more active at a particular time when more regulation of carbohydrate is required, or it will be less active and less of insulin is required. So now there is a certain gene present elsewhere on the DNA. At some other place on the DNA, there is another gene present, and let us highlight it with this particular yellow color. Let us say this is a particular gene. Now this gene, the mRNA that this gene produces, the mRNA molecule that this gene produces, will give rise to a protein. And this protein has only one function. This protein goes and acts on the DNA where the gene, it acts on the insulin producing gene and either stops it from working or tells it to start working. So such a gene, now I'm talking about this particular gene, such a gene which regulates the function of other genes are called regulatory genes. And all the other genes which are not regulatory genes are called structural genes. I hope this particular idea is also clear to you. Now we'll move a little bit further into this particular topic, and that would be the process of transcription in prokaryotes. Now we are going to look into the process of transcription in prokaryotes. Excuse me. Now here in this particular image we have two strands of DNA, and so this is one of the strand, the 5' 3' strand, and here we've got the anti-parallel complementary strand, the 5' 3' strand going in the opposite direction. Now we must remember that out of the two strands, only one of the strand will act as a template. Now what do we mean by this? The template is the strand which is used by the enzyme RNA polymerase. Now, we'll just recall about the process of replication. DNA, when it has to make its own copy, it makes use of an enzyme called DNA polymerase, a kind of an enzyme which makes polymer of a DNA. Now here mRNA or tRNA tRNA, or we got rRNA. These are RNA molecules. These are RNA polymers. These are ribonucleic acid polymers. Now they will require a different enzyme, and that enzyme is known as the RNA polymerase. Now out of these two strands of the DNA, so let us say this is the strand of the DNA, out of these two strands of DNA, so we have one here and one here, only one of the strand will act as a template, or will act as a strand, the complementary of which is basically made as an mRNA. So this strand, which is used as a complement to generate mRNA, is known as the template strand. So here we have the template strand. The template strand is also known by another name. It is also called the antisense strand. Right? Why is it called antisense strand? We'll just have a look at it in just a teeny weeny little bit. Now the strand which is opposite to the template strand. Now this strand is being used by an enzyme called RNA polymerase. For what? It is being used by the RNA polymerase to make mRNA. The other strand is not used by it. Why is it not used by it? We'll just have a look at that in just a little moment. But the other strand which is not used by RNA polymerase to make mRNA molecule is called the sense strand. Now it's called the sense strand. This makes no sense, but that is how it is. The opposite strand is called the sense strand, probably for the reason that the sequence on the sense strand is exactly the same sequence as on the mRNA molecule. Now what do I mean by this is, let us just go a little bit closer. Let us magnify it just a little bit and go a little bit closer. So what do we have here? Let's use a marker. So here I have got a C which is complementary to G here. So basically these were the original base pairs, right? And T is complementary to A and G is complementary to C. So these are all complementary base pairs that have been split apart for the process of transcription. Now if you look at the newly synthesized mRNA molecule, you see the sequence on this mRNA molecule is exactly the same as the sequence on the non-template strand or the or on the sense strand. Have a look. So the first base is G and here it is G. The second is U and the second is T. Now these are the same bases. Oh, but you think T is not U? Actually, thymine is the base pair present in the DNA, right? So this is the DNA strand, and uracil is the corresponding base present in the RNA molecule. So in one way you can consider both of them as equivalents. So if you look, T is equal to U, and here again we got G for G, we got U for T, A for A, and so on and so forth. So if you look at the sequence of this newly synthesized mRNA strand, the sequence here, the base sequence here is exactly the same as that on the non-template strand, and probably for this particular reason the sense strand is called so. Sense strand is called the sense strand for this one particular reason. I hope this much is clear to you. I hope you've understood what is the meaning of the sense strand and what is the meaning of the anti-sense strand. Sense strand is also called the non-template strand, but because it does not act as a template for the synthesis of mRNA or other forms of RNA, whereas the template strand acts as the as basically the copying material, complementary copying material for the synthesis of new mRNA molecule or rRNA molecule or tRNA molecule. So I hope this much is clear to you. Now let us look at the next thing. What are the different components that are involved in the process of transcription in prokaryotes? So the first component that is required in the process of transcription in prokaryotes is an RNA polymerase, right? Because what will RNA polymerase do? It will take the ribonucleoside triphosphates and bring them together and form a phosphodiester bond between the two. So it will, let us say, connect uracil to guanine to another guanine, etc., etc. So it will make a long base chain. Now what is important here is that in DNA replication we used, what was the substrate used? The substrate used was deoxyribonucleoside triphosphate. But in the synthesis of RNA cannot use deoxyribose sugar. So the substrate here is RNTP or nucleoside triphosphate. Ribonucleoside triphosphates are the substrates here. So what are the different components that are required? So the first component is an enzyme called RNA polymerase, but specifically DNA dependent RNA polymerase. This particular polymerase cannot act in the absence of DNA. So this is called DNA dependent RNA polymerase. It is a multifunctional enzyme, which we will just see in a little while. This is multifunctional. This is way better than the DNA polymerase in a couple of aspects, and not so great in other aspects. But anyways, and what else are required? Now there are two factors. One is called a sigma factor and one is a rho factor. So what are these factors? These are nothing but proteins. So basically two types of proteins are also required in the process of prokaryotic transcription, the sigma factor and the rho factor. Apart from that, what is needed is a transcription unit. Now let us try to understand the transcription unit first. Now let us say this is a sequence of DNA, and in this particular DNA, let us say this is the 5' 3' strand and this is the 3' 5' strand. Now in this particular molecule of DNA, we'll have to change the orientation a little bit so that it becomes a little easy for me to explain. So let me have the 3' end here and the 5' end here and the 5' end here and the 3' end here. Now let us say this is a particular gene, right, which will encode for a particular RNA. Now in prokaryotes, where should the process of transcription start from? The process of transcription cannot start from anywhere in this circular genome. So if this is a circular genome, where should the transcription start? How will the cell identify that the gene is here? Now, in order for the cell to identify, or the component of protein synthesis or transcription, in order for the different components of transcription to identify where the gene lies, there is a sequence, there is a region called the promoter region. Now, promoter region, what is it? It is basically the sequence of bases. It is the sequence of bases on the DNA, and this particular sequence of DNA is called the promoter region. This region is identified by proteins, and these proteins, they go and bind to this particular region, and then they act like flags or pointers for RNA polymerase to come and join. So this region on the DNA, or basically on the gene of the DNA, is known as a promoter region. In front of the promoter region is the coding region. This particular region in front of it, in blue, will basically code for an RNA molecule. So let us just draw the RNA molecule. So this particular region will make the RNA molecule. It usually is the mRNA molecule, right? So we'll talk in terms of mRNA molecule. It can be rRNA molecule or it can be tRNA molecule also. Now what else is present? What are the other components? So if the process of transcription starts, it must also stop somewhere. The RNA polymerase cannot simply just run over it and keep making RNA forever. It has to stop somewhere, right? So the place where it actually terminates, which is, let us say here, this particular region, this particular sequence of DNA rather, here, this sequence of DNA has a specific base pair, has a specific sequence of base, and this particular region is called the terminator region, or simply terminator. Now, recapitulating all of this once again, in prokaryotes, what are the components required? A polymerase enzyme, the function of which is basically to synthesize the mRNA, rRNA, or the tRNA strand. A sigma factor or the rho factor, which are proteins, which basically identify the start point and the end point of the transcription site, or basically this transcription region of transcription, right? And then we require a transcription unit, which is basically the gene itself, and the gene has a start point, basically an identification point, that this is the place from where the gene is starting, and this region is called the promoter region. And also the transcription unit has an end point, which says, okay, only till here the sequence of mRNA contains the information. Beyond this, there is no information, and this particular region on the DNA on the DNA, you must remember, is known as the terminator region. So basically the third component is called a transcription unit, and the transcription unit is made up of three parts: the promoter, the RNA coding region here, this part in the middle, this is the RNA coding region, and third is the terminator, which tells the RNA polymerase to just get off and not make any more RNA molecule. And the last component that is involved in the process of transcription is known as are known as the ribonucleoside triphosphates, or RNTPs. Right, so they are known as the RNTPs. So we will just take a break here, and we'll meet up in the next session for our in order to continue